Hi, everyone. I'm Jyoti Patel, and I'd like to go ahead and get started. Sorry, I think we've had a couple of technical difficulties, but hopefully, a year into virtual meetings, we're getting there. So thank you again for taking the time to come to this LingMap meeting. We hope to make this interactive, so please use your chat function and ask us questions in real time, but we're really excited about these sort of next steps. I'm going to go ahead and start, and Dr. Herbst will be joining us at any moment now. So let's go to the next slide, please. So this is our agenda for the day. We're going to, again, just recap a little bit about what's been going on with LungMap as well, talk about the future. And then we have really some important updates regarding the incorporation of Foundation 1 CDX to the trial. Our exciting study that was just launched
I'm here.
with Dr. Pata, 1900E, updates on some publications as well as updates on drug selection and committee overview. Hi, Dr. Herbst.
Hi.
Welcome. So you're on. I
am.
welcomed everyone on. Yeah. And I thought we'd go ahead and get started. I'm happy to keep going and then catch up, or would you like to
Well,
talk?
we have a few slides that I was going to show.
Yep, so we just got to the agenda. So the next slide is yours if you want to advance, please.
Yeah, thanks. It was quite a event signing on. I don't know if anyone ever saw that old movie, The Net. I felt like that trying to get the computer to start in time. Lots of pressure. Well, listen, I'm glad we're all here and look at this wonderful slide. Thanks, Jyoti. And that's actually one of the big additions since we last met. Jyoti being the vice chair and coming to us from one of the other cooperative groups, so working very closely. You can see we have medical oncology chairs, and you'll hear from almost all of them today. David, Constantine, Tiziana, Joel, Marty, and Saima. And then, two very important committees. Leora Horn moved on to AstraZeneca, so Haas Borgai took over as our drug selection chair. Dave Kozino, who you'll hear about in a little bit, as our translational medicine chair. Very active committees. I want people to think as they're hearing everything we talk about today about what could you do with some of these data. There are lots of opportunities to mine these data, and I hope many of you will do that. Then we have our translational medicine team, Fred Hirsch and Phil Mack. And then, Mary, of course, biostatistical lead, along with Catherine, Ji Ling, and James. Next slide. So I guess, and that picture is very nice for a couple of reasons. First of all, look at that trio in the front row. That's Harpreet Singh, who is basically for the FDA. She handles all the lung work. And then right in the center there, you recognize Mary, and that's Ellen Siegel. But this was the last time I think almost the last time we were at a live meeting this year. That was in Washington right before the COVID hit. And we all have to get back to doing that again. But that's a retreat we had, and we're planning to do another one of those as soon as we can. Next slide, please. So here's the current schema, and the green are the active, the yellow are temporarily closed. We don't have any of those right now. Blue is in development, and the dark blue is completed. So I'm not going to go through the completed. Much of that was S1400, the squamous only part. The good news is a lot of these papers have been coming out, and many of you have been part of them. We still have open in green. We have two studies. We have selpercatinib, LOXO-292. That's about to transform. That's a single agent trial, 1900B for red fusions. We're about to, of course, this is all second or more line. We're about to do a chemo combo. That's in the works. Just last week, the first patient went on S1900E. You're going to hear about that. Amgen 510, the KRAS. On the right, you'll see that we have for checkpoint refractory disease, N803, which is an IL-15 agonist with pembrolizumab versus standard of care. That's in development as well. Next slide. And then on this slide, what you'll see is, we're still all over the country, and we've persisted despite the COVID and the other issues. You can see the total number of patients registered to either 1,400 or LungMap. LungMap has now exceeded S1400 in just a couple of years, whereas S1400 was open for 3 or 4. And you can see you can either screen at progressive disease or you can pre-screen. And look, the majority are taking the pre-screening option. And then you can see sub-study assignments. 938 have been assigned on LungMap. About 2/3 of them screened at PD, a third pre-screened, and then 273 were registered to the sub-studies. So we've been accruing quite well despite this very challenging year. Next slide. And then I'm just in my last few minutes just going to say this is truly a partnership. And I believe now it's even more of one than ever before. You know, at this meeting, we have representatives from all the different groups. In fact, the virtual meeting has made this even easier, ironically. But you can see the master protocol in the middle of the NCTN with the NCI, SWOG, FNIH, the Friends, the FDA, Foundation Medicine. You're going to hear a little update on some of the screening, and then all of the companies that we've worked with shown on the bottom. And I think my very last slide is just about the patient. You'll see that on this slide, this is all about patient and patient care. And when we get to the next slide, you'll see that we have some input from just showing that this trial is benefiting our patients. So I don't know why it hasn't advanced, but I'll stop there. And yeah, there you see it. It really, and you know, the amazing thing, if you look on the left, as it shows on my screen, that's Herta Chow at the VA. And the VINS, the VA has been a major participant in this, and that's something that Dr. Blanke, I remember when I first talked to him about 7-8 years ago about LungMap, was really interested in. So it's great to see that. And then we see Annie on the right, and there are many patients. And you know, in a time when we need to get profiling available throughout the country, we need to get drugs to patients, many of these oral drugs, LungMap is serving a very important role in medical care. So now, as I said, it's been great working with Jyothi. We've known each other for years. We actually have worked together at Vail. And so Jyothi, the floor is yours.
Great. Thanks so much, Roy. So, you know, you've been with us through a lot of, through, I think, a time in which there's been incredible advancements in lung cancer. And our hope is that LungMap is sort of leading the way and continues to be a trailblazer in this area. And so I want to talk a little bit about what we're doing and what we envision in these next months. Can we have the next slide, please? And so a couple of things just to set the stage. So we know that from the time that LungMap was first introduced to where we are now in 2021, that by and large, there is wide adoption of NGS in non-squamous tumors. And so as a standard of care, most patients are getting wide NGS testing. Certainly there are regional differences, but for most patients with adenocarcinoma, this is paid for and is easy to get. We also know that immunotherapy is now a standard of care in patients who don't have a mutation. And patients are either getting it as primary therapy alone or in combination with chemotherapy, and often are doing well for very long periods of time. And so sort of the uptake until we meet people in the second line or beyond setting can be much longer than it was historically. We've seen huge gains in blood-based testing. And I think the thing that we are sort of struggling with, and this picture sort of shows, is that we have ambiguous historical controls. Certainly our ability to develop studies and to look at what we think were response rates in a docetaxel treated population changes significantly. So it may be a person who's primary refractory to immunotherapy or who may have been on a single immunotherapy for a long time and is now seeing chemotherapy with very good performance status and minimal disease burden. And so really looking at how we think about statistical assumptions as we've developed trials has changed a lot. Next slide, please. So I think that what we hope to do with sort of this background knowledge is to really think about what are the most salient questions and what are the unmet needs for our patients. One of the biggest is that although many patients respond to immunotherapy, patients develop either primary or have primary resistance or they have acquired resistance. And really understanding and overcoming these mechanisms of resistance, I think, is a primary aim for many of us. Other strategies include, or other problems and other unmet needs include really identifying rational strategies to treat patients who have received targeted therapies for a long time and now have a multitude of mechanisms in which to develop resistance. These tend to be smaller populations. Patients often have acquired mutations, and certainly these secondary and tertiary mutations occur in minorities of patients. And so collaboration across multiple sites is absolutely important. And then finally, how do we really integrate biomarker analysis, not only from the tissue as we've always done with NGS, but also in real time as response to treatment from blood? And how can we do that at point of care? Next slide, please. So as we think about sort of this future of lung map, certainly, this is from Roy's slide, and he told you a little bit about what we anticipate next. Go ahead, next slide, animation. So now what has changed? One, you'll hear from Dr. Neal about enrolling with a commercial Foundation 1 CDX. So we anticipate many of our patients with non-squamous tumors and an increasing number of patients with squamous tumors will have had commercial assay and can enroll at any time. We're also including cell-free DNA at enrollment and during study to look for early response. Next animation, please. We're looking at trials in TKI refractory disease with rational combinations, including multiple TKIs and anti-VEGF combinations. Next slide. As well as really thinking about novel immunotherapy trials and deciding whether or not we can, how to really look at refractory and patients with acquired resistance. Next. So if we think about this next generation, I mean, remains a very highly collaborative group. We want to do cooperation between cooperative groups, the NCI and industry, and we want to move quickly. It's important and incumbent upon us to really bring the next generation of researchers into this fray. And so with this effort in really getting group chairs and strong representation across all cooperative groups, we're hoping to really help junior investigators find a place to do a lot of these phase 2 trials. And then our ultimate goal is that this remains patient focused and centered. This is at the point of care, and we can see what an impact that made, particularly during COVID, where patients who were on LungMap continued to have access to some of these trials across the country when travel to tertiary sites was often limited. Next slide. So with that, I'm going to hand over to Dr. Neal, who's going to talk about Foundation 1 CDX testing. Thank you.
Thank you, Dr. Patel, and thank you, Dr. Herbst. Can everybody hear me?
We can.
All right, wonderful. I'm ducking out of a clinic workroom and going to do this. We'll spend a couple of minutes talking about the use of Foundation 1 CDX, which we've been eagerly anticipating, and then I'll be happy to take a couple of questions and then have to run back to see a few patients. So the Foundation 1 platform has been integral, as we know, to the LungMap study from the beginning. And we use Foundation 1, if we can go back a slide, I don't think I'm advancing these. Thank you. So the Foundation Medicine platform that has been the DNA next generation sequencing platform from the beginning of LungMap. And really that company has taken their platform from one of many DNA next-gen sequencing platforms to now FDA-approved companion diagnostics, which is widely used across the country as one of the standards of care. We've been aware since that CDX approval that there are patients that may have known CDX testing and prior testing, but we haven't been able to use that information to make them eligible for LungMap because of a variety of logistical issues. So over the last year, the LungMap chairs have been very interested in allowing CDX testing that's known without an additional tissue submission requirement. And I'll talk to you for a couple of minutes today about the mechanisms to do that. So this, of course, requires known testing as well as tissue availability. But for those patients, and we go to the next slide now, for those patients who have had CDX testing all the way back, dating to and including September 1st, 2019, now we can use that in lieu of tissue. So we anticipate the protocol that's currently in amendment, version March 1st, 2021, that should be released shortly, will allow either the submission of tissue for biomarker profiling, as is the current state, or a note and a consent that patients have had prior commercial Foundation 1 CDX testing results after September 1st, 2019. For those sites submitting the commercial results, they have to have the report available, and the sites will provide the test order. They'll provide results done on solid tumor tissue, so this does not include the Foundation liquid platform as of this point, and patients also have to consent specifically to have the commercial results released to SWOG. As one of the side effects of this, we realized over the conduct of the study that PD-L1 testing, which was included historically in the testing together with Foundation medicine testing for tissue on the lung map sub-studies, PD-L1 testing has been less important on the current sub-studies. So because we're not requiring tissue submission from everybody, we're not going to perform universal PD-L1 testing on that tissue any longer. But we do acknowledge that most sites probably have known PD-L1 testing results from tissue already when enrolling patients. So we'll just request that through the EDC. These commercial test results, you can't just say, here's the result, can we get sub-study assignment? We're actually going to do a repeat analysis of those commercial test results. So Foundation Medicine is going to take the raw data, re-put it into the analysis pipeline, and there is a chance that results can be different from what's on the commercial CDX report. So if we note that, or if the sites note that, that's because of the repeat analysis. So say somebody in 2019 had a gene that was noted that was a variant of uncertain significance, or VUS. It could be possible that now in 2021 that VUS is of significance and is noted differently. But this reanalysis will go into the same exact mechanisms to assign to sub-studies as new tissue testing will. So that is the reason that we will predict, at least at the beginning, up to a 16-day turnaround. This won't be instantaneous, even though the data is already there. Next slide. So mechanistically, what do sites have to do? And we'll go through this in a bit more detail now. For patients submitting commercial Foundation 1 CDX results, it'll go through the same STS system. They'll have a packing list. These details will be in the protocol, and it'll go through a metadata rave. And back one slide, thank you. So, and submitting all the details about the information to submit are down below in the bullet points and will be available in the protocol as well. Next slide. So in conclusion, the next protocol amendment, we hope, will allow prior tumor Foundation CDX testing, and we're very excited about this. We don't think that September 2019 will be very limiting, because most of these patients within the last year and a half or 2 years that have test results, they will be allowed to use this mechanism. And I just wanted to let you know that the study chairs are also looking into incorporating other DNA next-gen sequencing platforms as well. It was a complicated enough process just to use an alternative pipeline for the Foundation Medicine platform, but we hope to use the NCI match as a model in which other CLIA-certified labs can be approved for sub-study assignment, maybe over the next year or more. So we're working on that as well, so that patients don't need repeat testing and can get fast and immediate access to these exciting sub-studies within LungMap. Questions? Thank you.
Hey, Joe. Joe?
Yes. Yeah.
When do you think we will be able to use some of the other tests? How long?
Yeah, I think it would be hard to anticipate less than a year from now, but we're starting those discussions. We're forming a subcommittee in order to look at the mechanics of this. And I guess the question is, will we be able to at least incorporate 1 or 2 other testing platforms at the beginning and then have a mechanism by which other labs can apply for approval. I'm hoping, I think realistically it's going to be a year or more though, Roy, but I hope that we can have that to go as fast as possible. Okay? I know that there's an unmet need out there.
And Roy, just to remind you, we're going to try to go through the presentations and have a panel question and answer at the end.
Okay.
All right, that's great.
right.
We'll have to drop off to run to patients, but appreciate all of your attention today and support of this study.
Thanks, Joel. Well, now we have Jessica O'Donovan from our Site Coordinators Committee, Consent Form Changes and Implementation. Hi, Jessica.
Hi, everyone. So I'm just going to go through briefly over some of the consent form changes now that we are going to have the changes that were just talked about. Next slide. So first, for best practices of implementation and training, you're going to notice with this update that there are some very different things in the consent form that I'm going to go over after this slide. And it's going to take some very clear definition and probably a couple of presentations at meetings, and everybody who's consenting is going to have to be very clear on what they're saying to patients, because all of the information for every group for pre-screening versus screening is going to be on the same consent form, as well as previous testing and patients who need to send in tissue for testing. So pretty much, you're going to have to review the protocol very carefully. I would suggest bringing it to team meetings to go over a couple of times. And then prior to consenting, reminders of all the changes that have happened. And make sure that everybody is informed of any changes, of anything that's been removed and or added. Next slide. So the major changes. Previous commercial foundation testing, as we just learned, is going to be allowed for sub-study assignment. Patients will now have to consent to either releasing their testing through the study or to release previous results from Foundation 1 testing. And then they will still have to do the typical pre-screening versus screening, and all of it's going to be on one consent form. So that's really what I want to talk about, is what the consent form is now going to look like, because it's going to look very different. Next slide. The major change you're going to see first are what I'm going to call or statements. So for example, you'll notice up here on the upper left of my screen, I'm not quite sure which way it's oriented towards your screen, but if you have previous genomic profile test results, we are going to ask you to provide these to the study team, or we are going to ask you to submit a tumor sample to test for specific genes. So all of this is going to show up on the consent form. So you're going to have to make sure that patients know which one they're consenting to, because they might get confused and think that they're consenting to all of this, that they're sending tissue and previous results. And we just want to make sure that when they're being consented, that they're very clear on what they're consenting to. So you're going to see various or statements throughout the consent form. Next slide. So this section will appear under the what exams, tests, and procedures are involved in this study. And here is where it'll actually explain in depth what the patient is consenting to. So what will happen if they're submitting previous profile testing results, and then the typical what we have done in the past where we're submitting a tumor sample to the study team. So just make sure you go over which one applies to the patient that you are consenting at that time. Next slide. And then highlighted here is something we have added to the signature page, which is a major change and is very important. This is where they will actually be stating that they are agreeing to release previous profile testing results, and that they are authorizing Foundation Medicine to release and disclose any information performed to the SWOG group to use these results as to the study. So this is basically just how it's going to go through the system. We just need to make sure that everybody has permission to access everything they need to. So this will be added to the signature line of the consent form. Next slide. So these are just my major take-home points. I know this was very brief, but you know, there's probably more in-depth training at another time. So know which group your patients are consenting to. Again, pre-screening versus screening, and then previous testing versus tissue sample being sent. Make sure you read the statements that correspond with whichever situation your patient fits into. Make sure that patient knows what they are agreeing to, that it's one or the other and not both. Review the new consent form with all members of the team, potentially a couple of times to make sure, send out reminders if you need to, and make sure that all members of your team are aware of this exciting new change. I think this is something that we've all been kind of looking forward to, and I think it's a great change for the study as a whole. Thank you.
Okay, Louise.
Thanks, Dr. Herbst and Jess. I'm just finishing up the sequence on our change to allowing reanalysis of commercial Foundation 1 CDX results. And I'm going to do a very quick how-to with the logistics of this process. And again, we will have more training opportunities in the future, and you can always contact me for more questions. So this will be a little bit brief, but just to give you an overview of how it will work. All right, next slide, please. So here's a little visual overview. The squares in blue are the steps that the site will need to take. The squares in gray are back-end steps, and then of course the green is our end goal. So I'm going to go into more depth on the blue squares in a minute. You can see that we will have a RAVE form for uploading the commercial CDX report, but I just want to highlight that that does not actually request the use of those results for subsidy assignment. That request is done through the SWOG specimen tracking system, almost as if you're submitting like a dummy specimen, but actually no specimen will actually need to be shipped. So we'll go into those steps in a little more detail. Next slide, please. The first step will be during open registration to step 1 of lung map, the screening step. As part of your eligibility, you'll confirm that there's a commercial Foundation 1 CDX report that meets the requirement if you're planning to use this option. And then during the step 1 registration, you'll answer an auxiliary registration question regarding which approach will be used for biomarker profiling and select the reanalysis of commercial results option. Next slide. So next, you will navigate to SWOG specimen tracking system. This is just as if you, similar to as if you were going to ship a tissue specimen, but instead you will select this option here that's called request, and it will be going to lab number 242, which is just like a dummy lab that is for Foundation Medicine commercial NGS requests only. Next slide, please. This is a very important step. This is during the SWOG specimen tracking system process. You will answer what we call specimen-specific questions. In this case, these will be questions that are used by Foundation Medicine in order to link the request to their existing results. And so you'll see here, for instance, we definitely need the ordered test number that comes from your CDX report. We need some information about the patient, and we need the date of tissue collection and site of the original tissue that was used for the commercial results. Next slide, please. So lastly, in specimen tracking, you will need to select the button that says ship this shipment and generate packing list. In this case, there's actually no need to physically mail or email anything. The generating the packing list will act as the request to Foundation Medicine in the back end of the system, like electronically. So you just want to save that packing list for your records. It acts as like the requisition form, but nothing actually needs to be shipped. All right, next slide. So once that is done, the reanalysis will be going on at FMI, and in parallel, the site can submit the PDF report in RAVE. This will be in a commercial NGS testing folder that will roll out in RAVE, just like your other baseline forms. This will be used for source documentation only, so uploading the PDF here does not request the use of results for reanalysis and subsidy assignment. That is done through the previous process I just outlined in the specimen tracking system. Next slide. And finally, the end goal, the site will receive the sub-study assignment following the same process and timing as tissue submissions. So sub-study assignment will be sent to email contacts that are entered either during registration or on the notice of progression form. The sub-study assignment will also be available in RAVE. There will be no PDF reports attached to these emails. The PDF reports would have been previously shared via the commercial process, and if any updates occur during reanalysis, these will be shared to the original ordering physician using the same commercial process as well. And the timing currently is expected to be the same as the tissue process, so within 16 days after submission of the request, although we anticipate this will likely be shorter because this is a new process at FMI where they asked us to use a longer turnaround time window to start off with. And again, for patients pre-screened prior to progression, you will get a confirmation email saying that the results have been successfully transferred to SWOG, but the sub-study assignment itself will be sent after submission of the notice of progression in RAVE. Next slide. This is my last slide, but maybe the most important one. If this is especially for any site coordinators and nurses out there, if you are planning to implement this and submit commercial Foundation 1 results, please don't hesitate to reach out to your data coordinators, myself, Leah, and Amy. Our contact information is at the bottom of the slide. It's also in the protocol, and we would be more than happy to go over this process with you on the phone the first time or by email. We love to get questions ahead of time or even during the process because we can help you do it correctly the first time. That being said, if you think you made a mistake, definitely contact us as well, and we can usually help with that as well. So thanks so much, and I will turn it over to the next presenter.
Thank you so much. I think now we have Suki, and we have to welcome Suki to SWOG. She moved institutions, so we started this trial before you moved, but we're very happy to have you here and have this trial open. So Suki.
Thank you, Dr. Erbst. My name is Suki Pata. I'm at Cedars. The co-chair of this study is Dr. David Gerber, who's at UT Southwestern. And this study is examining AMG 510, otherwise known as sotorasib, in patients with KRAS G12C mutated non-squamous lung cancer. Next slide. So I'm going to give you some background on why this study is relevant. We'll discuss the drug sotorasib, the disease, KRAS G12C mutated lung cancer, also focusing on the diversity and heterogeneity of KRAS mutated lung cancer. And then finally, we'll conclude on discussing the schema of the sub-studies. Next slide. So sotorasib is a first-in-class KRAS G12C inhibitor, and KRAS G12C mutations are a very relevant target in lung cancer, affecting 13% of our patients. And how this drug works is that it specifically and irreversibly inhibits KRAS G12C by locking it in its inactive GDP bound state, which you can see on the schema on the right. Next slide. So this is the sotorasib phase 1 clinical trial schema from CodeBreak 100, which included a dose escalation portion and a dose expansion portion. And of course, the primary endpoint of the study was safety. Next slide. So what did we find out about the safety in the study? Well, first of all, there was no maximum tolerated dose. The recommended phase 2 dose was 960 milligrams daily. And majority of the toxicity were gastrointestinal in nature, including nausea, diarrhea, and then also elevated liver enzymes, AST and ALT. Next slide. I think this part of the study was one of the most exciting to see this in a phase 1 study. These were 59 patients with lung cancer treated across dose levels of sotorasib with a median follow-up of 12 months. And what was seen is that the objective response rate for this patient population was 30%. The disease control rate was approaching 90%. The duration of response was respectable at close to 11 months, and the median progression-free survival was 6 months. So this was very exciting because now we're seeing a potential avenue for an oral targeted therapy for these patients that did not otherwise have this as an available option. Next slide. So then there was a phase 2 clinical trial to understand what we saw in the phase 1 trial was going to be recapitulated in the phase 2 trial. So this included 126 patients with KRAS G12C lung cancer who were previously treated with treatment. And the primary objective endpoint was response rate. Next slide. So this is a waterfall plot from this phase 2 study. This is with a median follow-up of 12 months. And you can see that efficacy results were very similar to what was seen in phase 1. Objective response rate, 37%, disease control rate, 80%. And many of these responses were quite deep, as you can observe in the waterfall plot. Next slide. The other thing to know about this drug is the median time to objective response is fast. It's at the first assessment at 1.4 months. The median duration of response was again similar to the phase 1 study at 10 months, with 43% of patients remaining on treatment. And the progression-free survival was also very similar to the phase 1 study at 6.8 months. So now this is really generating a lot of excitement. Next slide. So what about the toxicity? Were there any new toxicity signals? The answer is no, not really. There were still GI toxicities. Toxicities were the most common with diarrhea and nausea. And then you just got to watch out for the AST and ALT increase, which in some instances can be grade 3. 7% of patients did need to discontinue treatment due to an adverse event, while 22% needed a dose modification. Right? So based on this data that you have just seen, the FDA did grant priority review to sotorasib in this indication of pretreated KRAS G12C non-small cell lung cancer, and the expected decision date from the FDA is in August of this year. This is very rapid drug development and very exciting for this group of patients who experience this type of lung cancer. Next slide. So we know KRAS mutated lung cancer is evolving as a very diverse disease, not only on variant subtypes such as KRAS G12C, but also with the co-mutations that follow KRAS mutations. So as you can see on the pie chart on the right, the most common co-mutations that follow the KRAS driver mutation are in what we call tumor suppressor genes, so P53, STK11, and KEAP1. And we know from very elegant preclinical studies that there are differences in the signaling of these subsets, the immune features, and metabolic programming, and that these co-mutation subsets may have prognostic importance for our patients. And I think as the treatment landscape is constantly changing, they also have potentially predictive value for our therapeutics. Next slide. So we have been learning some data about how these new novel KRAS G12C allosteric inhibitors perform in co-mutation subsets. And it's important to note that these are exploratory in nature. So this is looking at sotorasib. This is in 104 patients where they looked at the STK11 and KEAP1 co-mutations. And I think what is most notable is that tumors that contain KEAP1 mutations, whether or not STK11 was mutated or wild type, had an inferior objective response rate, while patients who just had STK11 mutations alone had similar probability of response to the overall population. Next slide. And there's also the similar data that's been reported with another allosteric KRAS G12C inhibitor, adagrasib, which is a compound by Mirati. And they examined co-mutation subsets in about 51 patients. And what was most notable here is that if there was a P53 or KEAP1 mutation, there was a similar probability of response to the overall population. In the 14 patients with an STK11 mutation, there was particularly a signal of enhanced response. But remember, these are small numbers and exploratory in nature, and sort of looking back at the study that has been conducted. Next slide. So this is that is the background of why we are conducting S19 we want to essentially evaluate prospectively how this novel allosteric KRAS G12C inhibitor sotorasib performs in co-mutated subsets of KRAS G12C mutated lung cancer. So the eligibility and complications, obviously with the KRAS G12C mutation, importantly, this is non-squamous lung cancer. They have to receive at least one prior systemic therapy. That would be chemotherapy alone, immunotherapy alone, or a combination of both, and no uncontrolled brain mets. Depending on the biomarker assigned from Foundation Medicine, patients get allocated to one of the cohorts, co-mutations, P53 mutations, co-mutations SWK11, and then finally sort of catch all buckets. So these include patients with tumors with SWK11 or P53 double co-mutations, or any co-mutation in the KEAP1 axis, because we know that's a bad actor. All cohorts receive sotorasib at the recommended phase 2 dose, 960 milligrams daily, until disease progression or intolerance. During this treatment period, it's very reasonable in terms of clinic lab and visits every 3 weeks in accordance with the cycle, and imaging is conducted every 6 weeks. The primary endpoint of the study is objective response, and then secondary endpoints include duration of response, PFS, OS, and safety. And I think one of the interesting things that we'll learn from the study is that we are collecting circulating tumor DNA. And from AACR, we just recently heard about novel resistance mechanisms to similar G12C inhibitor, adagrasib, which are quite heterogeneous, including novel KRAS mutations, which may switch to binding pocket, which is where the drug is going. So I think we also have a lot to learn about resistance from the study in these very co-mutated subsets. Next slide. So as I mentioned, the primary endpoint is objective response. The P53 cohort and the other cohort will target 40 patients in each of those cohorts. So the SWK11 cohort has a slightly lower sample size of 25, because we expect the estimated percentage of that study population to be lower. This is a two-stage design, so we will be looking at efficacy in a two-stage process. All the cohorts are designed with a 90% power, 5% one-sided type 1 error to rule out a response rate of 14%. And we picked that 14% based on historical studies with those Taxols, so our middle of the range objective response rate. Next slide. So obviously, this was a huge effort. As you can see, the lung map piece is huge. This is truly a collaborative experience. These are all of the names that participated in. Our lead statistician, Roy Norman, is getting us sort of to the finish line with the protocol, along with many other key players, including from the Amgen team. Next slide. So S1900 is open and available to your patients, and as you heard from Dr. Earp, there is one patient who is now fully registered. Thank you.
Thank you, Suki. That was great. Now for a study in development, as mentioned earlier, I believe it's going to be John Rengel presenting on behalf of himself and Hatem.
Yeah. Okay. Hey, can I? So this is S1800D. It's a phase 2/3 study of a combination immunotherapy versus standard of care chemotherapy or chemotherapy plus ramucirumab in the case of taxane plus ramucirumab. Next slide, please. The study is intended to evaluate if N803 combined with pembrolizumab can overcome resistance, either acquired or primary resistance to anti-PD-1 therapy. We're enrolling to 2 cohorts based on the primary stratification factor of whether of best prior response to an immunotherapy containing regimen, meaning whether a patient experienced progressive disease as their best response to treatment versus stable or partial or complete response to an IO containing regimen prior to enrollment to this trial and prior to progression and enrollment to this trial. The primary endpoint for both cohorts is overall survival. If I could have the next slide, please. So This is based on work that came out of my laboratory. We developed this as an investigator-initiated trial and published the safety and preliminary efficacy data previously. Next slide, please. The N803 molecule is based on IL-15. It's in the IL-2 family of common gamma chain agonist cytokines. N803 is modified from recombinant human IL-15 in that it is administered in combination in complex with IL-15 receptor alpha. It's a slightly mutated IL-15 molecule, and these modifications essentially increase its activity by over 100-fold over native recombinant IL-15 administered by itself. The advantage of IL-15 compared to the member of this family of cytokines that we may be more familiar with as IL-2 is that it acts on CD8s and NK cells and the presumptive constituents of the immune system, which exert anti-tumor activity in the immune system, and with relative CD4 sparing. We won't get into the caveats of CD4 subpopulations that have anti-tumor activity, but just in broad strokes to paint that it acts on CD8s and NKs without activating a regulatory response through interaction with CD25, which IL-2 tenaciously interacts with, and it's this differential activation of various components of the immune system, which we believe is responsible for the dramatically improved therapeutic index, which allows us to administer this entirely in the outpatient setting. Next slide. This is an example of a patient, a 65-year-old PD-L1 70% patient who had a PR to carbopem-pem in the first line. It lasted about 5 months before she progressed. This depicts her best RECIST response from enrolling in my trial, dropping chemotherapy, continuing pembrolizumab uninterrupted, and adding N803. The patient is now actually closer to 3 years out with a far deeper and more durable response than her chemoimmunotherapy regimen. So this and other patients were the first evidence of definitive evidence of activity for common gamma chain agonists in non-small cell lung cancer. Next slide, please. This is a waterfall plot of some preliminary from the phase 1. There will be an update on phase 2 at ASCO this year from industry-sponsored trials as well, showing best RECIST response amongst refractory and relapsed patients. As you can see, there's a number of patients who experience stable disease and some target lesion volume shrinkage even amongst refractory patients, and a number of patients who experience partial response with addition of 803 at the time of progression. Next slide, please. This is a tolerable regimen in the outpatient setting. The most common side effects are injection site reaction. The 803 is administered subcutaneously, and patients can experience kind of red, indurated reactions at the injection site. Very typical symptoms starting several days after the first dose of 803 are fever, flu-like symptoms. The highest temperature I've seen amongst any of my patients is 102. A little bit of Tylenol goes a long way for this. I haven't had to admit anybody for these side effects. No capillary leak syndrome or cytokine release syndrome. Interestingly, there appears to be no augmentation of the typical immune-mediated adverse events that one would expect from PD-1 therapies, and certainly this has been a tolerable and feasible regimen entirely in the outpatient setting. Next slide, please. With this slide, Mary, if you're with us, I'll turn it over to you to describe a little bit about the study design.
Happy to. Thanks, John. All right, so this just summarizes the design that we'll be implementing within the S1800D study. As was previously described, patients will be eligible if they've previously received anti-PD-1 or PD-L1 therapy and experienced disease progression on or after that therapy. We'll be stratifying patients into two cohorts. Patients defined as primary resistance having had progressive disease on the prior therapy within 12 weeks of initiation, and acquired resistance, including patients who had progressive disease at least 12 weeks after the initiation of the therapy containing PD-1 or PD-L1. Within the primary resistance cohort, the design will be a standalone randomized phase 2 study. Patients will be randomized equally between N803 combined with Pembro versus standard of care, and the standard of care will be standard of care chemotherapy or docetaxel combined with ramucirumab. We'll enroll up to 130 eligible patients, and the primary comparison will be a comparison of overall survival between the two groups. The study will include interim analyses, so that there might be an earlier closure if we don't meet endpoints at that point. In the acquired resistance cohort, we'll be implementing a phase 2-3 design, which means that we could enroll up to 300 patients on that trial, similarly randomized equally between those two treatment arms, comparing overall survival between the arms. But this has the possibility of taking it all the way to phase 3 sample sizes, and if it meets its endpoints, could be taken to the FDA for regulatory approval in this setting. I think that's it for this description. So we'll hold any questions on this talk, but John and Hatim, thank you for being able to join this morning. Next slide. So then I'll just go over, we're shifting topics here very briefly to just talk about what we've done over the last year or so, or I should say 6 months or so within LungMap in terms of publications. And you can see a list here of all the studies that we've published or are working on publishing right now. So just listed in alphabetical order, S1400G, which evaluated talazoparib, a PARP inhibitor, in patients with homologous recombinant repair deficiency positive disease. That paper led by Taufika Wanakoko and Lauren Byers was published in Clinical Lung Cancer in January of this year. S1400K, which was a study looking at ABB399, an antibody drug conjugate targeted against c-Met, was published in October of 2020, and that study was led by Saima Wakar and Suzanne Arnold. S1400F was a study looking at durvalumab and tremelimumab to overcome resistance to prior immunotherapy. That study was led by Natasha Lal and Nair Rizvi, and we are working on getting that study published. It's a wonderful paper, and we're just about to submit that paper for publication. S1400I was our randomized phase 3 study evaluating nivolumab combined with ipilimumab versus nivolumab for immune checkpoint inhibitor naive patients in the second-line setting in squamous cell lung cancers. Study was led by Scott Gettinger and Luda Basanova. We submitted it to JAMA Oncology. We've gotten a few minor comments back, and we expect that paper to be accepted for publication very soon. And then a non-therapeutic trial that we conducted within the original 1400 series study was a study led by Josh Roth and Scott Ramsey evaluating the attitudes, knowledge, and expectations related to return of genomic results. And that study was published in JCO Oncology Practice in April, so this month, and just a couple weeks ago. And then finally, we submitted a paper, and it was published in October of 2020 to summarize the full first experience of S1400 that evaluated the LungMap protocol in squamous cell histology. And that paper was led by myself and all of the people on this on this panel mostly. And that was published, as I said, in October of 2020 in the Lancet Oncology. So we've had an incredibly productive last 6 months or so and expect to continue that. And we encourage you to look at those publications.
Thanks, Mary. It's been a good year for publications, but equally good for translational research. And Dave Cosino is now going to talk about the Translational Medicine Committee.
Thank you, Roy. I'm really delighted to talk about it. I'm really proud of this committee. Next slide, please. Its purpose being to bring together expertise in translational medicine to assist in biomarker selection and definitions and sample collection decisions for sub-studies under development, to review proposals for prospective and retrospective translational research, and to assist in analysis and publication of sequencing and other correlative science data. Next slide, please. So the membership of the committee represents the multidisciplinary nature of LungMap, where we have many of the scientific leadership of the LungMap group together with FMI and Nationwide Biobank coming regularly to these meetings to discuss how to bring these different pools of talent together to answer TM questions. Next slide, please. And areas of expertise are in many areas, including therapeutics, particularly in immuno-oncology, targeted therapies, DNA repair, and anti-angiogenic therapy, but also in other areas critical to TM, such as biospecimen banking and processing, pathology, and clinical genomics. Next slide, please. And so in addition to some of the activities I discussed, we're always trying to make the most of the rich TM resources from LungMap. And so just as a couple examples of areas we're working on now, we've realized that slides stained by FMI, including most PD-L1 stain slides, and some H&E stain slides when FMI did not receive one from the site, that we've been sitting on 800 some of these slides and have an opportunity to use these for further analyses. We've also had residual extracted DNA from ctDNA submissions and have about 500 such samples that we're inventorying right now to see what value we can have from these samples for other experiments. And then not all resources are biological. For example, the images that are of the H&E and PD-L1 slides are stored digitally on an Apereo imaging system and provide a resource for such things as future algorithm development that could be accessed by the group. Next slide, please. And so, we want to hear TM proposals, really to utilize patient specimens gathered on LungMap clinical trials to improve outcomes based on specified laboratory criteria, what we think of as biomarkers. And these TM ideas can be broadly proposed by companies, by study chairs, and LungMap collaborators, including FMI. Next slide, please. And in evaluating these proposals, we really want to address the following questions. Now, how would this proposal lead to new important scientific information that will directly impact patients or indirectly by leading to future clinical trials? We ask, does this TM fit appropriately within the LungMap platform and the priority of other LungMap TM? We want to hear about sufficient background data and justification for the proposed science and understand specimen availability, including whether there is sufficient quantity of samples for the science to be conducted. If serial samples are needed, will a patient be excluded if they're missing any samples? And in looking at retrospective analysis linked to outcome data, we would need to know something about whether the primary manuscript is published or soon to be before we can discuss accessing those samples. If adding TM post-activation, we can look into whether retrospective submissions will be requested. And then, when applicable, how results might be provided to the treating investigators and patients. Next slide, please. And so I wanted to highlight some of the work that has recently been presented at World Lung including this very nice analysis of tumor mutational burden by next-gen sequencing in the two immunotherapy trials, S1400I and A, that Fred Hirsch is leading. This was a very nice oral presentation at that meeting. And some of his key conclusions included the following: that high TMB was associated significantly with overall survival and progression-free survival across our two IO studies, and that this was independent of PD-L1 status. A number of TMB analyses were conducted, including continuous and cutoff categories. Appeared that the continuous TMB variable analyses were generally more informative. The combination, interestingly, of TMB and PD-L1 expression did appear to impact outcomes in S1400I, where data on both were available, and how, therefore, genetic alterations associated with high TMB contribute to clinical outcomes would warrant further consideration. Next slide. Just as a sampling of the data, here is where the PD-L1 and TMB analysis was looked at in combination. And as one might like to see, the overall survival was greatest when PD-L1 was greater than or equal to 50%, and TMB was greater than or equal to 10, represented by the green lines that you see here. Next slide, please. I've been really excited to sift through the S1400 data because it really represents a wonderful resource. In fact, as near as I can understand, it's the largest NGS data set of advanced squamous cell lung cancers of previously treated patients, although it's not whole exome as such. It's limited to genes sequenced on Foundation One T5, the research platform, but nevertheless, a wonderful data set for analysis. And as one example of our interesting findings, we saw that mutual exclusivity of PARP4 and two genes, NFE2L2 and KEAP1, that have been known to be a part of responses to oxidative stress and are implicated in treatment resistance. These are both mutually exclusive with this third gene, PARP4. The next slide, please. And you can see visually how this looks overall on the left, where patients with tumors that have an alteration in KEAP1 tend not to have them in NFE2L2 or PARP4, and vice versa, that if you have a mutation in PARP4, you wouldn't have a mutation in KEAP1 or NFE2L2. And what does it mean? Mutual exclusivity could be due to many different reasons, but it may sometimes suggest that these three proteins work in somewhat similar ways. Which is curious because we don't really know much about PARP4. And so it'll be a way to start understanding how this gene may actually have a biological role that's not yet understood in cancer and could then be a potential future biomarker in clinical studies. We've also looked at co-occurrence, and just as an example of the power of the size of the S1400 data set, you could see co-occurrence where you see overlap of, say, the blue bars. Very nicely shown here for P53 and CDKN2A. Such overlaps were observed also in the somewhat smaller TCGA data set. But given the number of patients you can see here on the table to the right, that we've found so many more co-occurring gene pairs that you would be able to find in TCGA, in large part because of the greater case numbers. Next slide, please. And as it's a third presentation made at World Lung by Phil Mack, this was very important, I think, for the future of lung map, where he looked at the concordance of ctDNA versus tissue NGS. And here, he did this very nice work looking at driver versus non-driver mutations. And the driver mutations in particular, which are expected to be present in all cell lineages, had very high fidelity between plasma and tissue, sensitivity of 83%. The non-driver alterations showed maybe what could represent more tissue heterogeneity. They were less concordant. But regardless, where this could be particularly important is that ctDNA could allow for enrollment onto lung map substudies. If we keep this in mind, that a positive finding may merit inclusion in a study, but a negative finding would likely be inconclusive. Next slide, please. And to get a flavor of how these data looked, you can see here that for the drivers, those are the ones on the left bar here, there was that about 80% concordance, where you saw the same alterations in both the tissue and in the ctDNA. This is my last slide, but just wanted to really thank everyone for the opportunity to work with the Lung Map Committee, and we want to help at all possible stages from the time we're conceiving study concepts for future studies and to understanding how we could make the best resource utilization from all the many different rich sources of data and biospecimens we have in hand. So thank you so much.
Thanks, David. That was fantastic, and I'm sure there'll be some questions with you for you in a little bit. Now we're going to talk about the Drug Selection Committee. Haas?
Thanks for the opportunity to talk to all of you. I'm Haas Borgai. I'm at the Fox Chase Cancer Center in Philadelphia. I am new to the Drug Selection Committee, but I'll take you through the steps that we have put in place. The purpose of the Drug Selection Committee is really introduction of new sub-studies and ideas, also facilitating discussion regarding various proposals that come to us, and finally review and approval of formal sub-study applications. On the left-hand side of this slide, I've provided the names and contact information of a few people that could be helpful in this process if you are thinking about submitting ideas for us to consider. Membership of the committee is consisted of senior leadership of the Lung Master Protocol, investigators, industry, consultants, and the NCI. Next slide, please. These are some of the key aspects of this committee. As far as how we get ideas, is basically all of the investigators who are involved at various cooperative groups across the country can actually submit ideas to the Drug Selection Committee. Some of these concepts and ideas are actually initiated within the committee, and of course our pharmaceutical partners can also initiate a discussion regarding potentially having concepts discussed. The initial qualifications, as you can imagine, is an investigational drug that's basically ready for a phase 2 evaluation. Drug biomarker combinations with adequate preclinical data supporting safety and potential efficacy would be quite important to us. For the targeted therapies, obviously, we want to see that association, and for the non-matched therapy in advanced and metastatic lung cancer, regardless of histology, we would like to see these concepts. Basically, think of these as protocols that are ready for phase 2 evaluation. Candidates are evaluated by the committee. Again, key members are investigators that are putting the proposal together and other investigators that you have seen so far. We have experts in biomarker and molecular targets from academia, NCI, and FDA. And we also have consultants from our industry partners who are not conflicted with the specific concepts. Candidates are scored based on the appropriateness of the target for the patient population that we have in LungMap, the preclinical data, and of course, PK, PD, and safety data. Next. This really is my last slide here. As you would imagine, with many other committees, we have a process for selection. This is outlined here. I'm not going to go through every single one of these items, but just to give you an idea that, again, these suggestions come in. We have initial discussions with a smaller group of investigators, followed by an informal presentation by the investigators who have proposed the idea, our pharmaceutical partners, to the internal Drug Selection Committee. From there, the next step would be a presentation to the full Drug Selection Committee. This process involves completion of an application, which basically provides us with a summary of all the preclinical data that we normally would like to have for evaluation of the proposals. There is a formal presentation followed by Q&A, and then there is a closed session and discussion regarding what we had just heard. We would then proceed with formal voting by all the drug selection committee members, and we use 6 different criteria for evaluation, and each criteria is scored on a scale of 1 to 9. Those criteria again are shown, target and appropriateness of it for non-small cell lung cancer, the drug biomarker sort of relationship and understanding, preclinical data, PK, PD, toxicity, and the available clinical data, again, as it pertains to patients with lung cancer. And then from there is a final decision of the vote that's sort of presented to the investigators and our partners in industry. Once that process is completed, then there is a kickoff call with regular conferences and a whole process to get the protocols ready and to undergo all the review process that the protocols have to go through to finally have a sub-study that's ready for enrollment. I'm going to stop my presentation here and be happy to answer any questions during the Q&A session. Thank you.
Thanks, Haas. Welcome to the team. Mariah.
Hi, everyone. I'm Mariah Norman. I'm a lung map project manager and protocol coordinator, along with my colleague, Laura Gildner at SWOG Operations Office. We have probably emailed with some of you in the audience about protocol questions and regulatory questions. I'll briefly go over our protocol development process. Next slide. So there's 5 main steps for developing a new treatment substudy for lung map. As Dr. Borgai discussed, the first step includes discussions between the pharmaceutical companies and the lung map study chairs and team during the drug selection committee process. Once the drug selection committee and company are in agreement to move forward with the study, concept development begins. The concept is basically an NCI form that outlines the scientific background and justification for the study. If it's a biomarker-driven study, there's discussions to establish the biomarker definition at this time. Then development of the protocol and consent documents starts as soon as the concept is submitted to CTEP for review, just to get a head start on the protocol while CTEP is reviewing the initial concept. Protocol drafts are reviewed by the study team, company, site coordinators committee, and the patient advocate. In parallel with the protocol, there are several other processes going on, including development of the budget, contract, data collection forms, and the study build. Next, CTEP and CIRB will then review the protocol and consent documents. As SWOG holds the IND, we are responsible for also submitting the study to the FDA. And then finally, the study is ready to activate and open to participant enrollment once we receive CIRB and CTEP approval, once the contract is executed and drug is at the NCI Pharmaceutical Management Branch distributor. Next slide. So since LungMap opened in June of 2014, we've developed 20 concepts. We've opened 15 studies to accrual, completed 13 studies, as you saw in the schema earlier, and we've worked with over 12 companies. So I think as we develop more studies and work with more companies, we're continuously improving our process to activate high quality studies in a quicker manner. Over the years, LungMap has really continued to be flexible, adapting to changes in the therapeutic landscape and study structure. Next slide, please. So here you can see a snapshot of what the development process looks like. As you can see, it's quite a complex process with various departments and teams working behind the scenes. Our study team has really learned from the challenges as well as the advantages that come along with the master umbrella protocol. Based on all of our collective experience, we've streamlined the process by learning, what steps can be done in parallel. As you can see, many of these pieces are overlapping here. We've learned what questions to ask early on to reduce any potential sticking points. And we've established open communication with all partners from the beginning of development. As Dr. Borgai mentioned, we have kickoff calls with the companies and biweekly meetings to keep the momentum going with protocol development. And I think all of this has really allowed us to shorten our average timeline to 9 to 12 months from concept submission to activation, and ultimately providing more treatment options to patients faster. Next slide, please. So feel free to contact me about any questions that you have about the development process, any regulatory questions. Laura and I are here for those. Here's a list of other contact emails from all the team members for various types of questions. That's all I have. All right, I believe we're ready to open it up for a Q&A session. I'll hand it over to Dr. Gandera. Go ahead, Dr. Gandera.
Excellent. And why don't we get the whole panel on the screen? And David, it's all yours.
Okay, thanks, Roy, and thanks to all our speakers. Anyone in the audience who wasn't familiar with LungMap already, I think you can see from these presentations that this is a unique public-private partnership. I should add, on Friday this week, we will be having a meeting with all of our industry partners from pharma, which is very important. Obviously, they provide the drugs that we are testing in these protocols, and that way we can get input from them. We do have a couple of questions that have already come in, so I'll start with those. The first is very clear-cut. It should be quick to answer, and that is an estimated time for activation on SWOG 1800D. So that is the immunotherapy refractory protocol that John Rangel talked about. So Mary or John, when might we anticipate that trial could be activated?
I believe that we're meeting our prescribed milestones. Laura, or if you're on, if you could help me sort of answer that question.
Sure. Can you hear me?
Yes.
Okay, so the anticipated activation date for S1800D is Q3 of this year, 2021.
Okay, that'll be a very important study for us because as you heard, since it is a non-match study, it will complement the trials that we have ongoing that are genomically based for biomarkers, and also is a big study because of the two cohorts of patients and the randomization. The second question that came in early on is more a clarification, I think, and Louise Heilemann, maybe you or some of the other folks can just reiterate about what our investigators will expect to see in terms of Foundation Medicine reports. For the patients that come in with the commercial test, obviously the institution, the investigator will have the full 45-page report. A lot of it, of course, is regulatory required. It's not necessarily information that we use, it's references and everything else. But with our investigational reports, they are abbreviated. So will that change, or is that going to stay the same?
I don't believe there will be any change in the reports for those that are produced by tissue submitted through the study. Like you said, the commercial reports will already be obviously in the hands of the investigators, and if there are any updates, they will be informed of those. But yeah, I think this was Dr. Arnold's question, so I'm happy to discuss with her offline. I don't think it it only goes one way from commercial into research, and I don't think we can have it go back the other way from research to commercial.
And I think there are a variety of reasons for that, because it is an investigation report versus a commercial report. But Suzanne, we can maybe handle that offline to talk about it further. Are there other questions? Are there questions from any of our panelists to others that maybe we think we need to clarify something? I think you could see from these presentations, we have a lot going on, and it's a great team to be working with.
David?
Yes, Roy.
I'd like to ask David Kozno if someone wants to get involved with some of the analysis of some of these data. You know, they're interested in DNA sequencing. They have some ideas for different analyses. How would they communicate that to you in the committee?
Yeah, absolutely. Please feel free to email me directly. My email is dkozono@partners.org. I could put it in the chat. We also meet monthly. I think our next meeting is on Tuesday, May 18. And so if you'd like to reach out to be placed on the agenda to discuss an idea with the entire committee, we're always happy to put these on our agendas for the monthly meetings.
Other questions? We have about 5 minutes left before our conclusion.
Well, I have another one if no one else does. David, I'd like to ask the audience and the panel, what is the hottest thing now? We need more sub-studies. So does anyone have an idea they want to bring up now, or any thoughts as to where we could find them, lab work, or perhaps just please spread the word after this meeting, because we really are, you can see Haas has a very vibrant committee. We are really looking, I know there's some industry people here, we're looking for good ideas, and we have, you can see many sites open and accruing.
So there are a lot of hot ideas, Roy. I'm not sure we can spill all of our beans for ongoing discussions, because some of these are confidential right now with us and the pharmaceutical companies. But when you can't answer a question, the best thing is to throw it back to the questioner. What do you think?
Well, I what I would think is, what I'll challenge the group for is, when we started LungMap, we were linking targeted therapies with molecular drivers. And you can see from the slides we showed, we asked a lot of questions, and unfortunately not many of them worked, but they were important questions to ask: PI3 kinase, FGF receptor, and so forth. Now we have the KRAS, and my prediction is that's going to be really exciting. We already know that there are some hints of activity there based on the different mutations. Now we'll validate it. But we haven't personalized immunotherapy yet. So I would think that what's going to happen is we're going to, in the next 4 or 5 years, and I think LungMap's going to be a big part of it, we'll find ways to personalize why a patient is refractory. Is it primary refractory? Is it acquired? Is there a lack of TIL in the microenvironment? Is there some negative regulator? And hopefully, using this mechanism, we'll start making that non-matched arm with immunotherapy more matched. That's what I would hope.
And Roy, I'll just echo that, because I'm thinking exactly the same as you, and I'll extend it further. And that up until now, although we have had a variety of biomarker-driven studies, the goal of the study relates to a new drug. I think there's also an opportunity for us, in partnership with companies that develop biomarkers, to match that with pharma in terms of new drug combinations, and to have a trial that's actually designed to validate a biomarker. And I think there are a number of opportunities coming up with new biomarkers in the immunotherapy field, for example, which could lend themselves to this IO refractory subset that we're increasingly interested in. We do have another couple of questions that are coming in. One, Suki Pata, are you still on?
I'm still here.
And the question is one I'm sure you anticipate, which is, what will happen when a drug approval might occur that would affect your study?
Absolutely. I think it's in a way a good problem to have if there's more therapeutics available for our patients. But yeah, of course, it is a little bit of a concern given the accrual timelines of our study. For example, the P53 estimated accrual timeline is somewhere between 1 to 2 years. The STK11 cohort, just because the prevalence of that exclusive communication is a bit lower, between 1.5 to 3 years. And you know, I think the last cohort, which is sort of a catch-all, ensuring that we get patients on the study, is 1 to 2 years, but I think that timeline will be faster. So I think, you know, one area that we may struggle with is STK11. So I think it'll just be a bit more effort on the investigators' part, and also I'm working with our patients to explain the importance of this study. But understandably, that is a little bit of concern for accrual, but great also to have an FDA approval in the space.
I would just point out that a lot. Go ahead.
It's Karen. I just think it would be important for physicians right now to go back through their Foundation Medicine reports and find those patients that are KRAS positive and see where they are in their current treatment. So I think that would be very, very helpful. That's typically what we do. We go back to our Foundation reports, and that's pretty easy to do, and start looking and checking and seeing where they are. They may not be due right now, but they could be due in a month or 2 or 3. And so you have that list available, I think would be really helpful. And then getting them on to the screening lung map component in a much more proactive and aggressive way, I think could be very helpful.
And Karen, you and I were thinking exactly the same thing, because if this drug is FDA approved, it will not be for first line, very unlikely. And so the opportunity will be there to put your patient on this trial and help us figure out about the subsets of KRAS that likely affect the underlying biology and possibly efficacy of the drug. So there will not be a loss there. And again, we'll have time with the pre-screening if the drug is only approved for subsequent lines of therapy. And one more possible question, just as we're ending right now. Actually, I think our time is up. And so someone is asking here about having a trial based on P53. In other words, I think that has been an area where ideas have been formulated. A few trials have been done for many years. But of course, because this is a tumor suppressor, it's a lot hard to develop a trial, you know, compared to an oncogene. But I think we'll put on our thinking cap for that. And Roy, do you want to make any final remarks before we conclude?
Only to thank you and the rest of the panel. This has been a great session. I feel that we have tremendous momentum. Don't forget the lung, the open general lung session that starts in about an hour. So you have some time, depending on your time zone, for a break or for some lunch. No, but this has been great. Thank you, everyone. And we can make slides available to you as well, so that you can use them in your institutions to educate those that couldn't be here today. Thanks, and see you all next time.
Bye.